Thanks to Carl Thummel for selecting this work by Sudeep Mondal, Preetha Guptan, Edward Osuansa, and I'm Utpal Banerjee. This study initiated our interest in understanding how the mitochondrion controls cellular functions. The mutation studied here is CoVA, or cytochrome oxidase subunit 5A, of the complex 4 of the mitochondrion. Quite unexpectedly, clones of tissues mutant for the gene encoding CoVA can be recovered under certain genetic backgrounds. They do not die, and these cells can differentiate, but after a few rounds of cell division, the cells are blocked in their transition from G1 to S phase during mitosis. We have since isolated many nuclearly encoded genes that function in the mitochondrion, and they all share these characteristics. We realized that mitochondrial dysfunction triggers a checkpoint control in mitosis. Analyzing this further, in this paper and then in a subsequent work, we found that low mitochondrial activity will cause change in two different metabolites. First, shown in this paper, high AMP levels cause activation of AMP kinase, and in a P53 and archipelago, which is an F-box protein in Drosophila, dependent manner, causes specific degradation of cyclin E activity, thereby blocking the cell cycle during the G1 to S transition. Shown in a subsequent paper, mitochondrial defect induced increased ROS, which is reactive oxygen species, and this causes activation of the Jun kinase pathway and upregulation of the cyclin inhibitor DECAPO, which is a P27-like protein in Drosophila. So by two completely independent means, the cell blocks its G1 to S transition using mitochondrial metabolites as signaling molecules. Mitochondria are not just for generating energy. They interact with cellular functions in many different ways. And conversely, it is now becoming clear that many cellular functions directly influence the biogenesis and networking of mitochondria. Thank you, and congratulations to Developmental Cell.